Hello, everyone. A very warm welcome to our inaugural session to Voices Beyond the Slide Genetics to Diagnosis the Inside Story of Hereditary Colon Cancer. I'm Dr. Prachi Gupta, pathologist and the founder of Patho, and I'm truly honored to host this session. Today, we bring together patient experiences and expert insights to explore familial adenomatous polyposis and Lynch syndrome. We are honored to have a wonderful panel with us, Mr. Dan Shockley, who will talk about his powerful journey of living with attenuated FAP, Dr. Prerna Chadda, consultant oncopathologist from Rajiv Gandhi Cancer Institute, New Delhi. She will present the key histological features from Dan's case. Dr. Hema Kini, Professor of Pathology from Kasturba Medical College, Mangalore. She'll share a pathologist perspective on FAP and Lynch syndrome. Miss Rachel Woden, she's a certified genetic counselor from Stanford University. She'll talk about the role of genetic counseling, common hereditary syndromes, and importance of genetic counseling. Our first speaker is Mr. Dan Shockley. We are going to begin with our first talk. And our first talk is joined by Dr. Hema Kini, Professor of Pathology at Kasturba Medical College, Mangalore. She will share a pathologist's perspective on FAP and Lynch syndrome. Over to you, ma'am. Ma'am, can you unmute yourself?
Okay. Good evening. Can you hear me, Prachi?
Yes, ma'am.
Can you raise
And
your
are you
hand?
able to see my presentation?
Yes, ma'am. Yes, ma'am.
Wonderful. Good afternoon or good evening to everyone, whichever zone you may be present in. I would like at the onset to thank Dr. Prachi for including me into this webinar, a very, very interesting and fascinating topic indeed. Cancer, as we know it, is a disease of the genome, which results because of accumulation of multiple DNA perturbations that cause differential cellular survival, meaning that the cells can proliferate without being controlled, can survive even in the worst environments, and they are capable of spreading to distant sites and living even in those distant sites. Now, all this DNA disturbance results in what we call genomic instability, meaning acquiring and tolerance of multiple errors in the genetic makeup. This can affect genes which control the cell proliferation or inhibit it. So this results in genomic instability and inactivation of some of the tumor suppressor genes. Now, cancer as a whole can be sporadic, meaning that a person can acquire mutations later in life and develop the malignancy, or they can have an inherited predisposition because of some of the genes that they inherit. About 1% of all human cancers are inherited syndromes. And we find that most often the DNA errors are localized in the tumor suppressor genes, which normally inhibit cell proliferation, as well as in the DNA mismatch repair genes, which are responsible for repairing any errors that our DNA acquires. These genes are a group of genes which have several functions like cutting off the denatured segment or the damaged segment and replacing it with a correct DNA sequence. When an individual inherits a mutation in one allele of a pair of genes, that is called a germline mutation. And that person will be in a heterozygous state, meaning will have one abnormal and one normal gene when they are born. But with time, they acquire a mutation in the other uninvolved allele, and therefore the tumors that are formed are homozygous because both those genes are now having some DNA damage. So germline mutations are usually imparting a heterozygous state, and loss of this heterozygosity when the second gene gets hit is resulting in homozygosity of the tumors. Now, coming to colorectal carcinoma per se, this malignant tumor of the large intestine and rectum is the third most common cancer and is the second leading cause of cancer-related deaths. 80% of these tumors are sporadic, but 20% are familial. The understanding of the mechanics involved in the sporadic cancers came from the understanding of the genes that are damaged and the mechanisms involved in the hereditary colon cancer syndromes, the most common of which is Lynch syndrome and familial adenomatous polyposis. Now, when do we recognize a colon cancer as hereditary? If the individual or his family members show an increased risk of developing colorectal carcinoma with or without polyps in the colon. Usually, these tumors arise at a younger age than the sporadic colon cancers. Multiple family members are affected. The patients may have multiple polyps as well, which may be concurrently present or sequentially appear. They have other organ tumors as well, which help recognize these syndromic forms. Hereditary colon cancer is divided into three broad categories. Non-polyposis is where we find no polyps are seen or very few polyps, which are almost the same as what we see in the general population. And the classic syndrome we see here is Lynch syndrome. This is the most common hereditary colon cancer. It is acquired by an autosomal dominant mode of inheritance. It affects the mismatch repair genes which are listed below. They are a group of genes, as I told you earlier, which include MLH1, MSH2, MSH6, PMS2, and now EpCAM as well, which silences MSH2. Most often, these individuals develop colon cancer, but they can develop tumors in all the other organs that are listed here. The second most common hereditary colon cancer is the familial adenomatous polyposis syndrome. Again, this is an autosomal dominant disorder. The affected gene is the APC gene. Here as well, we see multiple polyps as well as carcinomas involving the colon, duodenum, stomach, as well as tumors in other organs like pancreas, thyroid, liver, and CNS. There is an attenuated version of FAP where we don't see as many polyps as we see in the classic FAP syndrome. Other syndromes which are rarely encountered are the MAP, the PAP-AP, and the HMPS, that is the mixed polyposis, the protease polyposis, and the mute-Y associated polyposis. Hereditary colon cancer is a risk in patients having hematomatous polyps as well, and this includes the Peutz-Jeghers syndrome, juvenile polyposis, and the Cowden disease. Let's look at a few features of Lynch syndrome. As I told you, this is the most common hereditary colon cancer and accounts for about 2 to 7% of all colon cancers. The incidence is about 1 in 279 persons. It is autosomal dominant in inheritance, and there is a germline mutation in the mismatch repair genes. When tested, if we find that the gene proteins are not expressed, we say that the person is deficient for MMR. If the genes are, the protein is expressed, we say proficient for MMR. Most often, if we look at all colon cancers, we find that MLH1 is mutated in almost 90% of the cases, and usually this MLH1 is associated with sporadic colon cancer. So therefore, the patients who have MLH1 deficiency will have to be further tested to see if there is a germline mutation before it can be considered a hereditary colon cancer. On the contrary, mutations of MSH2, PMS2, MSH6, and EpCAM are considered to be germline mutations. In sporadic cancers, it's more often hypermethylation of the MLH1 promoter, which causes the deficiency. Rarely, germline hypermethylation can also occur. Now, in our DNA, we have certain sequences which are repetitive and they are called microsatellites. These areas in our DNA are highly prone to mutations and are periodically checked and corrected by the MMR genes in normal situations. Therefore, if the MMR genes are mutated, we find that there will be DNA damage involving the microsatellites as well. And there are ways of testing the microsatellite. So when there is damage in the microsatellite, it's called microsatellite instability. In such of those cases where they do not find any damage in the DNA there, it is called microsatellite stable colon cancer. So Lynch syndrome is deficient in MMR and microsatellite unstable. Most often the carcinomas occur in patients around mean age of 45 years. The sporadic cancers usually occur at a mean age of about 60 to 69 years. We find that the tumors are more common in the right colon and have an exophytic or polypoid morphology. These tumors are not associated with increased adenoma formation. And we find that in such of those cases where there are adenomas, the progression to carcinoma usually occurs in a period of just 35 months. That is about 3 years time. Whereas what we see in adenoma carcinoma sequence, the progression from adenoma to carcinoma in the sporadic cases takes about 10 to 15 years. These patients can develop synchronous or metachronous colon carcinoma. The morphological types which we see that are peculiar to Lynch syndrome include the mucinous adenocarcinomas, the signet ring cell adenocarcinomas, and the medullary adenocarcinomas. Other morphological features that are clues to the possibility of Lynch syndrome include the presence of tumor infiltrating lymphocytes, Crohn's disease-like inflammatory infiltrate in and around the tumor. And contrary to the dirty necrosis, which we see in the usual colonic adenocarcinomas, there will be no dirty necrosis here. If the patient has a positive history of Lynch-related tumors, either in himself or in family members, that in addition will help us suspect the presence of Lynch syndrome. The tumors may occur in any of these organs: endometrial carcinoma, small intestine, gastric, ovarian, pancreatic, biliary, urethral, or pelvic carcinomas. Some brain tumors are recognized in these patients, and some patients with Muir-Torre syndrome have sebaceous adenomas and keratoacanthomas. The Amsterdam criteria is usually used to diagnose Lynch syndrome. Amsterdam criteria 1 is associated with the presence of colorectal carcinoma. Amsterdam criteria 2 refers to the presence of Lynch-related tumors. So usually we follow the rule of 3, 2, and 1. More than 3 relatives with either colorectal carcinoma or Lynch-related tumors, at least one of which is in a first-degree relative. 2 consecutive generations are affected by colorectal carcinoma, or if one member of the family has colorectal carcinoma at an age less than 50 years. The revised Bethesda guidelines are also used to diagnose Lynch syndrome. So patients who develop colorectal carcinoma younger than 50 years of age, presence of metachronous or synchronous colorectal carcinoma or Lynch-related tumors, presence of a colorectal carcinoma with MSI high histology, microsatellite instability high histology, And I told you the morphological features earlier. In a patient younger than 60 years, if we see these features, or if we see colorectal carcinoma in a first degree relative with either a colorectal carcinoma or a Lynch-related tumor in an age less than 50 years, or two or more first or second degree relatives with Lynch-related tumors at any age, then the risk of the patient having Lynch syndrome is high. Once we suspect Lynch syndrome, we submit tissue immunohistochemistry to detect the mismatch repair gene protein products. So when we stain the slide, if we see absence of staining, it means that the gene product is not there and therefore deficient. If we find that all these stains are positive, that is, we see the staining in the slide, then we know that we are not dealing with Lynch syndrome, and we stop with that. If we find that MLH1 and PMS2 are absent, then the next step would be to test for BRAF analysis. If BRAF is positive, then likely we are dealing with a sporadic colorectal carcinoma. If we find that there is no BRAF mutation and the history is suspicious of Lynch syndrome, then germline testing is asked for, and that in turn will confirm the presence of Lynch syndrome. If we find that there is no suspicious history for Lynch syndrome, then we test for MLH1 methylation. And if that is present, again, it points to sporadic colorectal carcinoma. If it is not present, then highly likely it is a Lynch syndrome, and we go ahead with germline mutation testing. So this is how we would approach a case with Lynch syndrome. Familial adenomatous polyposis is characterized by hundreds to thousands of adenomatous polyps carpeting the surface of the colorectal mucosa. It can involve the proximal GI tract as well. Here, the mutation is in the APC gene. This is a tumor suppressor gene located on chromosome 5. When this gene is mutated, beta-catenin, a molecule in the cell which induces proliferation, is not downregulated. So APC gene would inhibit beta-catenin, but here APC gene is mutated. There will be upregulation of beta-catenin that in turn induces proliferation in the colonic mucosal cells that results in the formation of multiple adenomas. However, with time, multiple additional mutations occur, including the KRAS mutation and TP53 mutations, which convert those adenomas into dysplastic adenomas, intramucosal carcinomas, and finally, adenocarcinomas. In FAP, the polyps begin appearing in late childhood, adolescence, and can be seen even in young adults. They may occur synchronously or metachronously. Adenocarcinomas appear by mid-30s. Most of them by the late fourth or early fifth decade start developing adenocarcinomas. There's no gender bias here. The frequency of FAP is about 1 in 8,000 to 14,000 cases. Besides the colorectal carcinomas, these individuals are also prone to tumors in other organs like carcinoma thyroid, pancreas, brain tumors, and hepatoblastoma. The attenuated form of FAP, the mutation occurs in the 5' or 3' end of the APC gene. Here, the number of polyps are much less than what we see in the classic FAP syndrome. Usually, the proximal colon and rectum are relatively spared in these patients. And sometimes we see there is late onset of adenomas and carcinomas. Extraintestinal manifestations, formerly this was referred to as Gardner syndrome, include osteomas, these are bone tumors, epidermal cysts, desmoid fibromatosis type tumors which involve the mesentery and abdominal wall, dentition problems, and congenital hypertrophy of the retinal pigmented epithelium. There is a high frequency of duodenal and thyroid carcinoma in these cases who have these extraintestinal manifestations. Turcot syndrome usually refers to hereditary colon cancer with CNS tumors. Now, this can be seen both in FAP and in Lynch syndrome. In FAP, the tumors that are usually seen are the medulloblastomas. In Lynch syndrome, it's glioblastoma. Mute YH polyposis is usually an autosomal recessive inheritance where there is biallelic mutation of the mute YH gene. And usually we see the adenomas about 10 to 100 adenomas. Individuals affected are 50 to 70 years and show a slightly increased risk of developing colorectal carcinomas. So in FAP, usually, most often, it is the tubular adenomas that are encountered. Some amount of tubulovillous and villous adenomas can also be seen. Early in the course, we find the formation of polyps with low-grade dysplasia. This then progresses to high-grade dysplasia. Finally, we will see intramucosal carcinoma developing within the polyps till finally it becomes an invasive adenocarcinoma. So for the diagnosis of FAP, we need to keep in mind multiple polyps, more than 100 in number. Family history of FAP with adenoma at an age less than 30 years. Family history of FAP with extraintestinal manifestations, as we had discussed earlier, and detection of germline mutation in the APC gene. In patients with a history of more than 10 adenomas, a family history of adenomas, or extracolonic FAP-type lesions, we advise that they undergo further genetic testing. Germline APC mutations can be seen in both FAP and AFAP, that is the attenuated FAP. Usually the region in which the mutation occurs differs for those two lesions, and a biallelic germline mutation of the mute YH gene is seen in MAP. So this is how we diagnose the familial adenomatous polyposis. So to summarize, hereditary colon cancer diagnosis can be very challenging because the lesions and the malignancy can appear at a very early age. Attenuated phenotype may not reveal all the features. Extraintestinal lesion information may be lacking. Family history lacking in de novo germline mutations, and therefore further testing is required to detect these cases. And basically, the study and the research that has gone into these mutations has given valuable insights in our understanding for gastrointestinal neoplasms. So pathologists would first correlate the clinical history and the pathological features. Such of those cases where we suspect hereditary colon cancers, we would flag the potential cases for further testing. Genetic counseling is recommended, especially it's beneficial for the index proband and families when first detected. And the pathologist's role will continue in surveillance endoscopies for biopsies that are done for subsequent lesions that appear in these patients. Thank you.
Thank you, ma'am, for your detailed overview on FAP. Just a quick note, if anyone have any questions during the session, please write them in the chat box. We'll address all the questions at the end of the session. Now, I'm going to introduce our main speaker, the voice of voices beyond the slide, Dan Shockley. He's a retired US Navy veteran and patient advocate who will share his powerful journey with attenuated familial adenomatous polyposis, a story of resilience, early detection, and hope. We are honored to have him share his story with us. Over to you, Dan.
Thank you very much. I'm honored for this opportunity. Like I said, my name is Dan Drydock Shockley. I'm retired Navy, served during Operation Desert Storm and during an Iraqi Freedom veteran, the campaigns. And I underwent my first and only colonoscopy I was just over 51 years old. I scheduled it with the Veterans Administration healthcare system when I resided in Hawaii. And the results revealed 100 polyps embedded throughout my colon, rectum, and anus. and I didn't have any symptoms, had no idea any of this was going on. So I was immediately referred to Triple Army Medical Center in Hawaii to the certified genetic counselor to have sequencing testing to rule out the APC mutation. And it took 6 weeks to get the results back. In the meantime, I had an open door with my genetic counselor, my colorectal surgeon, my GI doctor, the chief of surgery, and my primary care doctor. So I was able to gather information as much as I could about the type of surgery, a total proctocolectomy with a permanent ileostomy, if the results came back confirming I had the APC mutation in my fifth chromosome. So I wanted to read about the type of surgery, about the mutation itself, and then what it's like to be a permanent ostomate. So, I mean, theory is one thing, practical applications another. So, um, I served during my military time career. I served 4 different deployments to the Persian Gulf, so I'm well adapted to improvise, adapt and overcome adversity, as well as plan for the worst and hope for the best. So in this case, I went into battle mode in a positive way because I wanted to learn as much as I could, like I mentioned earlier. So the results came back confirming that I had the APC mutation, and it was in the best practice of medicine to have the TPC with permanent ileostomy surgery. So that was conducted 2 weeks after the results came back. And when I woke up, I knew where I was. I knew what I was there for, and I asked my nurse, the team there at the recovery room, if Dr. Gagliano, my surgeon, if he found everything he was looking for. And I find out that they removed my colon, rectum, and anus, and of those 100 polyps, one of them was causing an 80% blockage in my ascending colon. And come to find out, when the biopsy was the surgical pathology report came in a week later, it revealed that that large polyp was an 8 centimeter high grade dysplasia in my tumor in my ascending colon. So uh, Dr. Gagliano was able to remove all the associated lymph nodes, and once again, being asymptomatic, no family history, so no one on either side of my family knew anything about this. I'm the youngest of my siblings, youngest of my first cousins, so I share everything with them through the years. And um, now it's important to note, because I was 51 years old, that I've been diagnosed with attenuated FAP. The window for attenuated FAP is between 50 and 55. Now, uh, the late Dr. Henry T. Lynch, the founding father of hereditary cancer research, is credited with the discovery of attenuated FAP. He conducted his studies. He started in 1984. He received a grant from the American Cancer Society, and 6 years later led to the discovery of attenuated FAP being a subvariant of FAP. And as we know, it's an autosomal dominant germline mutation. Well, the the interesting part here was, once I had my surgery, I was notified by my genetic counselor that Dr. Henry T. Lynch was going to be coming to Hawaii to conduct academic lectures, and I was invited to one of his lectures at the University of Hawaii Cancer Center. And my genetic counselor, she introduced me to Dr. Lynch before the lecture in private, personally, and then afterwards we sat down, my genetic counselor and a few colleagues from Triple Army Medical Center. We sat down with Dr. Lynch, and for an hour and 50 minutes, we got to talk to the world-renowned leader of attenuated FAP. So I had my copy of my DNA test results, my pathology report, and he went on to say that I have a unique case of AFAP, because I was asymptomatic and no family history. So he said that about 1/3 of the diagnosis of AFAP are de novo. So he encouraged me to have my routine endoscopic surveillance, and he even offered to stay in contact with me. I could reach out to him. He said, I can share my pathology reports with him from the biopsies, and he will review it and give me his feedback and insight. So I did that through the years, and the last time I communicated with him was in September of 2018. And June 2nd of 2019, he passed away at 91 and a half years old. And he was, at the time, the director of preventative medicine at Creighton University in Omaha, Nebraska, among other things. And um, it's interesting because he passed away on Sunday, June 2nd of 2019, which is National Cancer Survivors Day here in the US. So how fitting, here we have the founding father of hereditary cancer research. He ends up passing away on National Cancer Survivors Day. So from that point on, my focus, my purpose in life is to educate medical students, share my patient's perspective about attenuated FAP to continue the legacy of Dr. Lynch on the importance of early detection, prevention, and hope is saving lives. And so I've reached out to numerous medical students and colleagues globally. I've done a live case presentation for the Tripler Department of Medicine, the University of Hawaii GI residency program, and recently I've been selected to be invited to be a live case presentation for the Stanford MS program in human genetics for genetic counseling, as well as the Stanford School of Medicine Molecular Foundations of Medicine course. So it's an honor to have that opportunity because I wanted to educate the medical students. Because I look at it this way, I was diagnosed when I was 51 years old. Dr. Lynch was 86 and a half years old, and my GI doctor, my only colonoscopy, my first and only colonoscopy, my GI doctor was 70 years old. And after my surgery, I was talking to my GI doctor, and he said, "Now, Daniel, I was going to retire 6 years ago." And I said, "Well, you know why you didn't retire, Doc? Because you had to save my life." So we had a good chuckle out of that. Now, 5 years ago, next month, my routine surveillance, and I undergo the surveillance at Palo Alto VA Hospital right down the road from Stanford. I relocated to Northern California because I knew the older I got, that I would be a candidate for surgery to spare my pancreas from the mutation. So 5 years ago, next month, we've discovered a 3 centimeter adenoma was discovered in my jejunum, and it was recommended that I consider surgery. And I thought that the surgery was going to be the Whipple procedure because I communicated with Dr. Lynch about that type of surgery years earlier. And I find out there's a newer medical technology in the last 25 years, and it's known as a pancreas sparing duodenectomy resection surgery. And there was a surgeon at Stanford who's one of the nation's leading pancreatic surgeons. So I was able to have that surgery completed. And I go about my life as if nothing happened. And it's just once again, then when I have the opportunity to share my journey with to as a from a patient's perspective for the medical students and the medical community, and I can do that globally now. We have virtual connectivity, and I take my journey, being able to adapt into this lifestyle, not just having the hereditary colon cancer syndrome, but also having a permanent ostomy. And I created an acronym for the word adapt, and that's attitude determines the ability for a positive transformation, because that's what my body's been doing. I had to transform it to be a permanent ostomy. And I like to take a negative and turn it into a positive. So I created a mantra for my life, and that's always forge ahead with a purpose. And my mantra is a positive spin on a bleak diagnosis. If you take the first letters of each of the words, always forge ahead with purpose, it spells AFAP. That's just how my mind works, and this is a way that I can, share my positive attitude, and with the audience. And I'm just honored for this opportunity. And the journey's been amazing. And I'm learning as I go. I continue to learn about the mutation, the different you know um, results from the endoscopic surveillance. So I undergo the surveillance now every 2 years, and I'm just, I'm a patient at the Stanford Cancer Center, for indefinitely. So I'm only about an hour and a half away from there. But I like to look at, like I said, take a negative and turn it into a positive, and so I'm able to, to take this my journey and share it globally. But I want to thank you for this opportunity, and feel free to ask any questions.
Thank you, Dan, for sharing your incredible journey with us and for reminding us the human side of genetic pathology. Thank you so much. Our next speaker is Dr. Prerna Chadda, consultant oncopathologist from Rajiv Gandhi Cancer Institute, New Delhi. She'll be taking us through the key histological features from Dan's case. Over to you, ma'am.
Thank you, Prachi. I'll be sharing my screen. So am I on full screen?
Ma'am, I can see your screen. If you select slideshow, then I'll be able to see your entire screen.
Okay, it's
Yes.
on slideshow, so I think
Yes,
it's
ma'am.
okay. Okay, so in continuation with what Dan has told us already, so I'll be talking about the gross examination of the specimen and the microscopic images for his case. So a total colectomy was performed, and this was the specimen received. Along with the total colectomy, there was also the rectum and anus resection. So towards the right, you can see this was the colon, and this was a large exophytic mass protruding into the lumen, and this was the one which is possibly causing some amount of obstruction for Dan. And the mass measured around 8 into 6.5 into 2.5 centimeters. So it was a big exophytic mass, and it was located in the ascending colon just adjacent to the ileocecal valve. So along with the large mass, there were also several other mucosal polypoidal lesions which ranged in size from 0.1 to 0.7 centimeter, while the rectum and anus also had similar sessile and pedunculated polypoidal lesions ranging in size from 0.1 to 1.2 centimeter in the greatest dimension. So these were the sections. On the left, the total colectomy sections, while on the right, we have the sections from the rectum and the anus resection. So this is a micrograph showing the large exophytic mass. You can see that there's an adenomatous proliferation of the wall, and yeah, so you can see there's some amount of crowding of the glands. There are elongated glands, and towards the right, you'll be able to see on the higher part, there's a nuclear stratification, and there is a decrease in the amount of mucus producing cells and the goblet cells, and you can see some mitosis here and there. And this appeared to be more villiform. This was another portion in the same adenomatous neoplasm. So this represented the villus portion. So in its entirety, it was called a tubulovillous adenoma because the villus portion comprised more than 25% of the entire adenoma. And predominantly, it showed a low-grade dysplasia, while a high-grade dysplasia can look something like this, where you can have cribriforming and marked nuclear pleomorphism with prominent nucleoli. And the other smaller tubular adenomas looked like this with crowded, elongated glands or crypts and with nuclear pseudostratification. So a final diagnosis was given out as a large tubulovillous adenoma, along with presence of multiple tubular adenomas dispersed throughout the colon as well as the rectum and the anus. Thank you.
Thank you, ma'am, for this insightful session on Dan's histopathology slides. Our final speaker is Rachel Hoden, genetic counselor from Stanford University. She will discuss genetic counseling in hereditary colorectal cancer, covering Lynch syndrome.
Thank you so much to Dr. Gupta for organizing this webinar and inviting me to speak. My name is Rachel Hoden. I'm a cancer genetic counselor at Stanford Healthcare out in California. I'm going to talk a bit about hereditary cancer risk assessment and genetic counseling, specifically for patients with colorectal cancer. This is outlining the different causes or etiologies of colorectal cancer. And as many of you are aware, there are multiple causes of colorectal cancer, the largest of which is what we consider sporadic or random chance disease. So that's outlined here in the red, where we all have a risk for developing colorectal cancer. And these are random chance events or mutations that occur in the tissue that can lead to a tumor developing. A small portion, this 5 to 10% that's outlined, is what we consider hereditary or inherited disease, where there's a single strong risk factor, often called a mutation or a pathogenic variant, and often inherited in families. So that predisposes an individual to a higher risk to develop certain types of cancer, including colorectal and often other types as well. The third type is familial. So this is often referred to as multifactorial disease, where there certainly could be some underlying genetic risk factors, but there's also other factors at play, either environmental or in some cases hormonal factors or other things that we don't fully understand. So I'm going to focus on this small portion of inherited or hereditary colorectal cancer. Hereditary colorectal cancer syndromes, there are different what we call phenotypes, so presentation of disease. I'm going to talk about kind of two broad categories. One are polyposis conditions, and I know Dan's going to share a little bit of his story if he hasn't already. And then also what we consider non-polyposis colorectal cancer risk that's inherited. Within these different types or broad categories, there's also multiple genes that can influence the type of polyp or the type of presentation for some of these different genes. Of hereditary polyposis colorectal cancer, there are multiple genes that can contribute to polyps of differing types, but oftentimes there's adenomatous polyposis. And so the very classic example of this is APC-associated polyposis, which presents as adenomatous polyps. And this includes familial adenomatous polyposis, FAP, attenuated FAP, and this syndrome called GAPS, which presents with gastric adenocarcinoma and what's called proximal polyposis syndrome. So all of those conditions are caused by pathogenic variants or mutations in this APC gene. This is typically what we see are presentations of many, many adenomas. There is a variability though. In very classic FAP, it's usually hundreds to even thousands of polyps in the colon and often in the upper GI tract as well, like the duodenum. And it's a very high penetrance for colorectal cancer. So in those cases, patients often need prophylactic colectomies or proctocolectomies, so both the rectum and the colon removed in often cases, depending on the burden of polyps in the rectum. And then this condition also can present with extracolonic tumors. So that includes upper GI cancers, including, again, the duodenum, osteomas, thyroid cancer, and then central nervous system tumors as well. Another very classic polyposis syndrome is a recessive condition. So it's caused by biallelic pathogenic variants in this gene called MUT1H. So both an inherited pathogenic variant from mom and then also an inherited pathogenic variant from dad. And so this biallelic presentation of variants, meaning the mut-YH gene, neither allele is functioning. And it can also lead to adenomatous polyposis with a pretty large range. Some patients have very few polyps, whereas others have such a polyposis burden that they also need a colectomy in some cases. And then there are some other more rare polyposis syndromes that can present with adenomatous polyps. I listed some of the genes there. And then also some hamartomatous polyposis syndromes where we see hamartomatous type polyps rather than adenomas, although there are some scenarios in which we see a mixed phenotype as well. Just given the time constraints, this other kind of broad category of non-polyposis hereditary colorectal cancer includes a condition called Lynch syndrome, which some of you may have already heard of. It is the most common inherited cause of colorectal cancer. About 2 to 3% of individuals with a colorectal cancer will be identified to have Lynch syndrome. And we know that it's also underdiagnosed. There's a lot of individuals who have Lynch syndrome who don't know about it, mostly because there's a wide range of presentation, and sometimes the family history doesn't look like a classic kind of Lynch family. Lynch syndrome also increases risk for non-colorectal cancer, so things like uterine or endometrial cancer, ovarian cancer, gastric cancer, and others, also quite gene dependent. There are 5 genes associated with Lynch syndrome, and they are genes that protect us from tumor development through this function of mismatch repair. And so when they're not functioning properly, most tumors will present with what's called mismatch repair deficiency or microsatellite instability. And so we're going to talk a little bit about kind of how we identify these cases, and of course, very relevant for our pathology colleagues who are often the first ones who identify that this could be a case of colorectal cancer caused by Lynch syndrome. And certainly elsewhere as well, there is a recommendation for what's called universal mismatch repair testing. And so this is tumor testing for mismatch repair deficiency on all colorectal and often all endometrial cancers, and sometimes all GI cancers and even beyond the GI tract as well, depending on the specific protocol at the individual institution. There's multiple ways to test for mismatch repair deficiency. A very common way to do that is actually the second one that I listed, which is immunohistochemistry for mismatch repair proteins. And so staining tumors to see if there is protein expression of MLH1, PMS2, MSH2, and MSH6. And often the patterns of the mismatch repair deficiency, let's say if there's a loss of staining of MSH2 and MSH6, we often will then understand better what the underlying causative genotype might be if this was a case of Lynch syndrome. Other ways that we can test are for, PCR-based microsatellite instability testing. It's often also done with a lot of these labs that do somatic sequencing of tumors. They will also perform MSI testing, of course, for treatment-related implications, but also for identification of patients who have Lynch syndrome. And whereas most people who have Lynch syndrome who develop a cancer will have mismatch repair deficient tumors, we know that many people will have mismatch repair deficient tumors in which they don't have a germline Lynch mutation. And so we're trying to differentiate and find those families that do have Lynch. Schematic and workflow of how we may handle additional somatic or germline testing in the scenario of a patient that has a tumor with a mismatch repair deficiency. We might utilize, of course, germline testing. There's also additional somatic testing that's often done. For example, tumors that have MLH1 PMS2 deficiency, we're often subsequently performing either BRAF mutation analysis or MLH1 promoter hypermethylation, because those are other etiologies for mismatch repair deficient tumors that are not germline Lynch. This kind of brings us to the next portion of my talk, which is putting it all together. So, if we do identify a patient with a mismatch repair deficient tumor, or they have a personal or family history that's suggestive of an inherited polyposis syndrome or an inherited colorectal syndrome that's non-polyposis, who is handling the follow-up for these cases? And in a lot of scenarios, there are access to genetic counselors like myself who would be able to work up a patient for suspected hereditary cancer syndromes, hereditary polyposis syndromes, and make sure that we're offering them appropriate germline testing, follow up on test results and interpretation, and then often following multiple other family members in the setting of inherited disease. So I kind of outlined multiple pieces of what we do as genetic counselors. We have advanced training in medical genetics and counseling, and really our role is to guide and support patients who have either a suspected or confirmed inherited disease. That could include cancer, could include other types of diseases as well. In the US, we're nationally certified, and there's different certifying bodies universally, globally. We, as I mentioned, interpret family history records, medical history, genetic test reports. We certainly provide education to our patients, and then counseling around what are the next steps once an inherited pathogenic variant or mutation is identified. How do we get other family members tested? And what are the psychosocial or psychological implications of having a genetic disease? Pedigree or a family history that we take to try to identify if the patterns are suggested of inherited disease. We would identify who has had cancer in the family and at what ages. So this would include, patients who have personal history of cancer, patients with family history only of cancer. We might also get referrals from a molecular tumor board because there's a somatic result on a test that would be suggestive of an inherited or germline mutation. So we might also get a referral in that way. And then there's often incidental findings where there might be a cancer-specific risk factor that's identified from a genetic test that was performed for a different indication. And these are the ways that we help guide patients through this process depending on how they present to care with us. Mention some of the specific benefits of germline testing. So this idea of identifying if there's an inherited risk factor. So a lot of times patients and even providers will ask, well, if the patient already has cancer, what's the purpose of understanding the etiology? And there's certainly multiple benefits, primarily, and even one that didn't get listed here, could be for treatment-related implications. So there are certainly genetic conditions and inherited cancers that are treated with certain medications and targeted therapies that would change an oncologist's recommendation for treatment of a particular cancer. And that could include, germline testing, and sometimes somatic testing, of course, would also clarify a specific treatment option. And then once an inherited risk is identified, one of the biggest benefits to the patient is allowing for condition-specific screening recommendations. So if a patient presents with an endometrial cancer and we identify that individual as having Lynch syndrome, then we would also recommend that that patient undergo more frequent colonoscopies for the purpose of early detection or even cancer prevention for a future colorectal cancer. And then in some cases, we are talking about preventative options like prophylactic surgery or risk-reducing medications. And then in the event that a germline or inherited risk factor is not identified, we can also provide families with empiric risk or observational data risk numbers to clarify what kind of screening their family members should have. So if there's an early colorectal cancer in the family, making sure that their close relatives are also getting sometimes earlier or more frequent colonoscopies. that beyond the individual patient is thinking about what's called cascade testing or family member testing. So I always describe as really the whole family is our patient. So when we identify an inherited risk factor, we can then offer genetic testing to other family members, often for, again, prevention and early detection. So oftentimes those are family members who they themselves don't have cancer, and we have great proactive opportunities to prevent or identify cancers at earlier stages for those relatives. This is a true kind of guidance across the lifespan. So thinking about testing and appropriate ages for testing. There are scenarios where we would test children. Other times it's very adult onset conditions. And so we're really prioritizing autonomy and letting an individual make their choice around timing for genetic testing. Of course, we get the benefit of what's called a true negative result. So there are some family members for whom they test negative for an identified risk factor, and then we can provide them, support around general population screening recommendations. And so that can certainly provide psychological relief, medical relief to eliminate the need for additional screenings that they might have been recommended had they not undergone genetic testing. And then, of course, proactive management, as I mentioned, for family members who test positive. There are lots of support resources, and I kind of highlighted some things that are specific to colorectal cancer risk. I think Fight CRC might be behind my video, Alive and Kicking, an organization called FORCE, the Collaborative Group of the Americas. So these are a lot of resources that patients have. Well, some of this is certainly US-based, but globally there's many different support groups that really focus on helping and supporting patients who have inherited colorectal cancer risk. Thank you for your attention, and again to the organizers for inviting me to talk. I'm certainly happy to be responsive to any questions that come up with my email there. And I always make a shout out to a wonderful scholarship fund that funds genetic counseling master's students who are underrepresented in our field. And so there's some information there in honor of a colleague and close friend of mine, Lauren Ryan. So thank you again so much for your attention.
Thank you, Rachel, for your comprehensive insights into genetic counseling. We have reached the end of our session. There's one question for Dan. Dan, are you there? We can't hear you, Dan.
He's on mute.
Okay, here we go.
Yes, Dan, the question is, what message would you give someone newly diagnosed with colon cancer?
Well, that's a great question. The first thing I would do is have a communication with your family members as well as your medical team, and to determine what the next course of action is going to be. So, if you have family history, if you're symptomatic, even if you're asymptomatic, just have a discussion with your family members, and then, get with your medical team, and then perhaps we can even get an appointment with a certified genetic counselor. And that would be my recommendation.
Thank you, Dan. And there are a lot of early career professionals and budding pathologists. Do you have any message for them?
Oh my gosh, yes. The medical laboratory professionals are the unsung heroes in the medical community as far as in my personal experience. I've had the opportunity to have an incredible experience at Tripler, because I actually got to meet the leading pathologist, and that's how I actually have the images of from my surgical pathology, and I actually have the 25 permanent section slides from the my total proctocolectomy surgery. So this is that started me early. Now I'm not a medical professional; I'm just a very attentive patient, and I just ask lots of questions, but I'm always, honored for the opportunity to share my journey to give back to the medical community, especially for the medical students and for the medical laboratory researchers. So I'm honored, to collaborate, and yeah, just it's amazing the things that go on, behind the scenes. No pun intended. So that's a good GI joke, right? So, but yeah, I'm just thankful to be able to share my patient's perspective because it's made a difference in my life. So.
Thank you, Dan. There's one more question. How do you see the colorectal cancer screening in future?
Well, it's going. The technology continues to advance. So this is the beauty too of early detection. And there's the Dr. Henry T. Lynch, being the founding father of hereditary cancer research. I mean, he started his research back in the '60s, and so perhaps in the decades to come, we'll continue to advance to be able to minimize the um, the cancer from reaching other organs, and yeah, so the medical technology as we continue to advance, I mean, we've come a long way. We got a long way to go, but we just make progress as much as we can, and I hope that helps.
Thank you, Dan. As we are running late, so I would like to conclude the session today. Thank you so much for all the speakers, Dan, Dr. Prerna, Dr. Hema, Rachel, for this wonderful, wonderful talk, for their time, for their dedication, for their expertise. And to all of you who have joined us today, thank you for being part of this important conversation. Thank you so much. And stay connected with us at Pathoscope for future sessions. Until then, always forge ahead with a purpose, like Dan says. Thank you all for joining us today. See you next time. Bye bye.